So I am Éric Karsenti. I did my PhD in Pasteur actually, and then in Paris, and then I went to California for 3 years, and I was in EMBL from 1985 to 2013, I think, something like this. I was so I came as a group leader in the cell biology unit, and then I became, when Kai Simons left, I took over the coordination of the program. I don't remember when this was exactly, but then I did this until the end. And this program, I tried to do a mix of cell biology and physics and biophysics in the unit. Yeah.
And how did you find your way to EMBL? Where did you first hear about the EMBL laboratories?
So actually, I did not know about EMBL. So when I was in San Francisco, I did not think of coming to somewhere else than Paris. I wanted to come back to Paris. And while I was there, actually, Kai, I don't remember if it was Kai, but somebody from the cell biology department called me. I think it was Graham Warren, actually, who called me. Once I got a phone call in the lab, actually, and it was Graham Warren, and he asked me whether I would be interested in coming as a group leader to EMBL, and at the time I said no. I wanted to go back to Paris. So yeah, so I came back to Paris, and then things did not pan out exactly as I wanted. So I called Kai back, and I asked him whether he would still be interested in me coming and trying to see whether we could work together. He said, yeah, sure, I mean, come. So I came, gave a talk, and it worked out. Yeah.
The rest is history.
The rest is history.
The rest is history. And in those days, in the mid-1980s, what was EMBL like? It's obviously, it wasn't as large as it is today.
No, yeah. It was a lot of fun, actually. I arrived here, and but it's interesting because it was like a bit like in California, UCSF. You know, everybody was very relaxed. I mean, working a lot, but very relaxed, and there was a sense of community. You know, collaborative and very supportive. Very supportive place. Almost no competition between people, so it was very nice. I really enjoyed it. I mean, you know, in fact, I did not want to stay very long in EMBL. You know, initially, I wanted to go. My wife was not so happy not to go back to France, so I thought maybe I come here for a few years and then we go back. But then I got so excited about working at EMBL, I could not leave. As I stayed for 25 years afterwards. Yeah.
I think that's a very common experience. People find that the atmosphere is so conducive to research, and you form collaborations, I'm sure, with many people.
Many people, yeah. I started to collaborate with many people. And the other thing is that technology was being developed all the time. So this was like, you know, I had the feeling it was like an organ, you know, you can pull the different registers and do many different things. So it was very extremely exciting.
Yeah, we overlapped for some years when I was a group leader at EMBL, and I know in my own case it was tremendously exciting and very useful to be able to use the micro-injection apparatus Ansgar developed, but also Ernst Stelzer's new confocal microscopes, which were completely new.
Exactly.
I assume for you also in cell biology, these would have been incredible.
Actually, useful. You know, I think we were with Kai, we were the first, and Ernst, we published the first confocal paper.
Really?
At EMBL on MDCK cells. So it was funny because we had Kai had hired a postdoc, Bacalao, it was his name, and he was in my lab actually. So he was working, trying to visualize polarizing MDCK cells with the confocal. And the confocal was really a prototype. So it was a huge machine and very sensitive to temperature. So each time we were trying to take a picture, the machine would go wrong. I remember sometimes Robert Bacalao was trying to do visualize things during the weekend when cells were ready, and the thing would collapse. We would call Ernst, who would come reset the machine and everything. It was really exciting, yeah. And there was something funny because, you know, we were not used to look at cells in 3D with confocal at the time. There was almost no software, very little software to do this. So the initial images, we were seeing dots in the cells, polarized cells, and Robert was telling me, but I don't understand this. Microtubules are fibers, they're not dots. And suddenly I told him, but you know, maybe because when they grow, the microtubules become vertical. So indeed, that's what happened. And then Ernst he started to do 3D imagings with this, and we started to see the cells from the side, and we could figure out that they were indeed like this. So this was all, discovering new technology and new, it was nice.
Very exciting. And you're also well known for your vision in bringing physics to address basic problems in cell biology. How do you see the role of physics in cell biology?
I think, there are different levels. Physics has been in biology for many years. The crystallography, the DNA, the structure of DNA, all that stuff, and proteins. But there is another kind of physics, which is statistical physics. And a lot of things in biology, in cell and developmental biology, have to do with collective behaviors. So molecular interactions, complex reactions inside cells, also in developmental biology, collective cell behaviors, different modeling, modeling dynamic systems, self-organized patterns, all that stuff. And this you need physicists both to do models, to do modeling, computer modeling, but also for the use of specific imaging systems like molecular interaction with FRET or things like this. And the physicists are necessary for this because it's kind of complicated to set up the machines and the analysis also. And so physicists are different level. I mean, the imaging, all the technology and theory. Yeah.
Yes. I mean, I grew up studying molecular biology and came into research where I think the philosophy tended to be a reductionist one, where you could reduce phenomena to individual interactions between 1, 2, or 3 components and study them in isolation in vitro. And if you did that on enough separate systems, you would understand examples, you would understand the whole system. And increasingly, I think that's just wrong, because there are these emergent behaviors where the combination of populations have behaviors that you can understand by only looking at individual components. And I think perhaps that's one of the things you can...
That's one of the things we can do as physicists. Well, this is a way of thinking of physicists which helps you to build experiments or to use the proper technical approaches to study large population of cells or networks, networks of interactions. And I think we need still to have parameters. Like, for example, it's very important to have enzymatic values of enzymatic parameter, reaction rates and things like that. So biochemistry is still very important because you get this reaction rate. But then you can use this to understand complex processes in the cell you are working with. This is what we call systems biology now.
No, I think that's probably one of the best definitions of systems biology.
Right, exactly.
Different people sometimes mean different things with systems biology, but cell biology really is systems biology.
Absolutely. If you want to do it properly, it's
And it's very exciting, I think, also where you see some fields of molecular biology and genomics and proteomics moving. So instead of only describing one component, you can actually measure almost everything at the same time.
Exactly. Now we can do that.
Yes, exactly. And that becomes very exciting for really driving this together. But of course, you moved on also to this wonderful project called the Tara Oceans Project. Perhaps you could introduce that and tell us a little bit about where that came from.
So I had, it's strange because I sometimes I made a short circuit, but the real thing is that I started to think about this by reading the book of Darwin, The Voyage of the Beagle. And the book is really interesting because it's 5 years, he spent 5 years on the Beagle around the world. And the goal of the Beagle was mainly to make the mapping of the south coast of South America. But Darwin went on this and he started to make a lot of observation. But he did a lot of observation, both scientific observations, but also it was like he described the adventure also. You know,
Yes.
he went horse riding in
Yes.
South America with gauchos.
Yes.
He tells all these things that he did in the, you know, in the Pampas and all this.
Yeah.
Since there were still Indians there. So the book was extraordinary because this was a mixture of adventure and science. So I thought maybe this would be interesting to redo this kind of adventure with a sailing boat. Initially, it was mainly to do popularization of science, so talk about modern cell biology, developmental biology, evolution, you know, during the trip at different places where Darwin was. But then we never managed to get this off the ground because, you know, nobody would fund a boat and an expedition like this just to talk about science. So finally I said maybe the best is to do a proper scientific expedition and then use this, you know, also to do communication. So that's when after, you know, foraging a lot, trying to find the right thing, I finally met the people from Agnès B, which is a fashion company in Paris. So Romain Troublet and Étienne Bourgois. And I told them, so the boat actually, they had a boat, they called Tara, which they were doing a very, very special expedition in the North Pole, you know, that put the boat, this is an aluminum boat, that was designed to be put into the ice in the North Pole. So that they made the drift again, the Nansen, you know, drift. And the boat was still in the ice when I contacted them, and I told them, you know, I would like to do an expedition studying biology, you know, in the oceans. And they said, yes, why not? That would be interesting. But they said, you know, the boat is still in the ice. We don't know in which shape it will come out. So this was in 2007, the fall of 2007. And then the boat came out from the ice in January 2008. And it was fine. So they said, okay, let's go. So that's how the whole thing started. And we planned, and actually we decided there were several possibilities. You know, we could study coral reefs because this was nice for media, TV and stuff like this. I was thinking also of deep sea vents because this is also interesting things. But corals were fine, but deep sea vents we could not do because Tara is too small. So there was another possibility, was to study plankton, because plankton is full of different organisms which are very nice. So I had a friend, you know, Christian Sardet, who always was interested in plankton in Villefranche-sur-Mer. And I started to talk with him in Villefranche and an oceanographer friend of him, Gabi Gorski. And we decided what we should do is just sample all organisms, you know, end to end, from viruses to fish larvae, around the world, in the surface waters, plus mesopelagic. So we decided to do this.
And this had never been done before?
Never had been done before. Yeah. And in addition, there was I think that until now, there was no technology capable of doing this. Yes. You know, either imaging or sequencing, not speaking of sequencing. And so I said, then I had another meeting with the people from Tara and told them we would do this. So then they took a guy to do films, and we started to talk about the whole thing, you know, do the science and the movies and all that stuff. And that's how it started. Afterwards, it was a lot of work because we realized that we needed to find scientists which were specialists of viruses, bacteria, protists, zooplankton, plus sequencing people, imaging people, bioinformaticians, all that stuff. So this was a lot of work. So we managed to find all these people. And then we had meeting. I got some 300,000 euros from the French ministry to organize meetings every 3 months for a year and a half before the expedition, and with people from different regions in the world, you know, from all over the world. So we had these meetings. This became really exciting because you know, making oceanographers talk to bioinformaticians, talk to people working on viruses and all that stuff, and And then we came up with a fantastic plan
Yeah.
which was the one we used for sampling.
Did you realize what a treasure trove of data you were going to generate?
Well, first at the beginning, I was very nervous because I thought, Jesus, I involve all these people, and if the thing fails, this is going to
How could it fail?
a disaster. Well, it could, because when we started, the sequencing was not up to the job yet. It was 454, and the first sequencing we did was pretty lousy. So, and then right away the Illumina came out, and then that was it. I mean, it worked. So then I knew it was fine. But
so this was the perfect timing, just at the point where it just became possible to do this.
Yeah. And the other thing which is amazing is that the imaging also. At EMBL we could do sophisticated 3D imaging.
Yeah.
And the high throughput also because of Rainer, this microscope where you can measure.
Yeah, with Rainer Peppercock.
Yeah, with Rainer Peppercock, with a lot of things. But also in Villefranche-sur-Mer, they were developing methods to recognize automatically the species using actually deep learning. So this was the beginning of
Which
AI.
is also, good timing.
It was just the beginning of AI. Yeah. Most people did not know about AI. And I started to use it on the boat actually with friends. I mean, with this guy from Villefranche. And it was amazing because the computer could recognize the species. So we had to train the computer with different pictures and things like this. But after that, it starts to learn by itself.
Yes.
So this was really amazing. And so there was a combination of high sophisticated technology development and this extensive sampling that we could do. Yeah, it's interesting because there was one American guy who was a specialist of single cell sequencing involved also. And one day he told me, this is amazing, this is science fiction. It was almost like, it was true, I mean, it was almost like science fiction, the experiment. And we were doing this on a sailing boat, and the 22 years old sailing boat. Yeah.
So it's an inspirational story, and I'm sure it's created, as I said, a treasure trove of data that so many other people can use and mine and build on. What would you say at the moment you feel was the most exciting discovery from this data set?
I think there are several things. I mean, the first thing is that this is the first time that we describe a complete ecosystem end to end, and for the whole planet, basically, because we sampled in all the oceans, including the Antarctic and the Arctic Ocean. So this is the first thing. The second thing is that this has allowed to discover millions of genes that were unknown. Yeah. And also the sheer number of genes, we were very surprised because we identified something like 160 million genes from viruses, bacteria, eukaryotes. I did not expect we would find so many genes. I mean, it was...
And the number of viruses also is
just mind-blowing. I mean, it's mind-blowing. It's mind-blowing. I mean, the number of viruses, big. And the thing is that we have been studying, RNA viruses, DNA viruses, gyruses, all that stuff. So, and I mean, we know things we did not know at all before this, So this is very exciting. Also, there are many things. I mean, because of the number of metagenomes, metagenomic. I mean, the number of genes we identified. Some people managed to reconstruct quite a lot of metagenomes of individual species, but also from single cells also, because we had kept a sample for single cells. So we have a large number of metagenomes which start to be used now to look at the complexification of genes and genomes during evolution. So that's very exciting. Plus, we also discovered very unexpected missing links between different types of viruses, early, very old viruses in evolution, both for RNA viruses and gyruses. Also, I think that it's already there quite a lot, but I don't remember now. But there are other things interesting. Yeah.
And a large amount, if not all of these data, are publicly available.
Yes.
Is that correct?
Yeah.
Yeah.
It's all the data are stored at EBI.
Yes. The
So
European Bioinformatics
European
Institute.
Bioinformatics Institute, run by EMBL, actually.
And we will put links in this video for people so that they can easily locate this.
Look at it. Yeah, I did not talk too much about imaging, but imaging also was, one of the incredible results also was the discovery of a huge number of interactions between microorganisms, protists, bacteria, inside protists, photosynthetic organism with heterotrophic ones that either are in endosymbiotic situations or exosymbiotics or parasitic, lots of interaction that we are starting to study now, but we knew this existed, but the extent of the diversity of this interaction was not known. So, and still not known because we still have to analyze this. So the imaging plus the genomics will be very useful to address these questions also.
I also have another project for you.
Really?
Yep, absolutely. So you've sampled life as we understand it, as it exists all across the oceans. How far does life go up into the stratosphere? How about sampling life and finding, are there any genes specific to microorganisms that exist at different altitudes?
Yeah. Actually, this is a good idea. Tara has done some of it. I think there was in recent expeditions. They had a capture up in the mast, and they captured some organism there. But it would be interesting to sample
much higher, because now we have
to sample
vehicles and we could sample.
Yeah,
this would be really interesting to see how, life came from the sea to land, but also explored the air.
Yeah, absolutely. Well, it's transported actually.
Yeah.
There are lots of organisms that are flying.
Yeah.
So it would be interesting to see how high life goes. Yeah.
And if there's any special adaptations of organisms not described yet by biology. So there's another project.
It's another project, yes.
Exactly. Are there any thoughts you'd like to share with us about your views of EMBL and its importance for Europe and for not just Europe but for world science?
Yeah, I think laboratories like EMBL are very important because you get people. First, you get extraordinarily motivated people who come here, and it's a very interdisciplinary environment, and it's very important for the world because to get people from different countries to work together. Well, this happens not only in EMBL because science is international, right? But here it's specific because people. Students, postdocs, group leaders come from all different countries, and we all are different, but together I think people are stronger because we're kind of a little bit different,
In a good way.
In a good way.
It's the good side of diversity.
Exactly.
Diversity is a superpower.
Exactly. And it's a real pleasure to, and I think it's very important because people go back to their countries afterwards, but they have this experience of having worked with other people from other countries. So I think this is a very important kind of mixing place. I think conceptually also. It's the environment is so supportive that people always develop new ways of thinking here. So I think it is very important, and it's not like EMBL, like probably other European institutions like ESA or the CERN. It's not the EU that is running it. It's really intergovernmental, and this provides freedom that you don't necessarily have in national labs. The Tara Oceans also is a very good example of it because I think the Tara Oceans, I would never have been able to take this off the ground if I had not been at EMBL.
Yeah.
Because I was, being at EMBL, I was protected from the local politics, in France or in any other country, And this protected us also from, you know, people, nasty people or anything.
Yeah.
You are kind of, it's a very strong protection against stupid politics.
Yes.
Yeah.
And of course, France is famous for, I think, inspiring the Médecins Sans Frontières.
Yeah.
But really, EMBL is Sciences Sans Frontières.
Exactly, yeah.
Forschung ohne Grenzen and Science Without Borders. And that's what we should go for, because the value of it. Éric, it's been great speaking with you. Thank you for coming and telling us about your career and your achievements and this wonderful Tara project, which is still going on. And I'm delighted that you've agreed to publicize even more with a new book, and also to go take Tara into space.
Yes.
Great.
Thank you.
Thank you very much.